This video is to help you with chapter 3.4 on the inheritance section of the genetics unit. All right, now we can't have a genetics discussion without talking about the father of modern day genetics, who is Gregor Mendel. And Gregor Mendel did a lot of work, but his most famous work was definitely on pea plants. So what he did is he crossed or bred pea plants with different characteristics, and he started keeping track of their offspring. And he did this in ridiculously large numbers, and he started to notice that some patterns emerged. And so because these numbers were so large and because the patterns were so consistent, he was able to make a lot of really important conclusions in terms of what characteristics are heritable or passed down from parent to offspring. It'll be much easier to have conversations with you about genetics if we can speak the same language and understand the same terms. So first I want to start off with genotype, and genotype is a symbolic representation, like the organism's actual genes. And so since an organism inherits 2 alleles for each trait, when we choose a letter to represent that allele and we write out both of them, that's the genotype. The phenotype is the physical characteristic that comes from that genotype, like eye color. Okay, so this would be the genotype. The genotype determines the phenotype. All right, a dominant allele is always going to be expressed in the phenotype, okay, because it's dominant. So if there are 2 dominant alleles inherited, of course they're going to show that dominant phenotype. But if something inherits a dominant and a recessive allele, it's still going to have the dominant phenotype because that's what it means to be dominant. On the other hand, this recessive allele is an allele that we're only going to see expressed in the phenotype when it's paired with another recessive allele. So in this example, since blue eyes is recessive, in order to have blue eyes, you have to have 2 recessive alleles for that trait. All right, now not all things follow that straight up dominant or recessive pattern. Some of them are what we call co-dominant. So if an individual inherits 2 different co-dominant alleles, then both of them are going to be present in the phenotype. So like a black chicken and a white chicken making a speckled chicken. Again, that only happens if they are co-dominant. Locus is a word that we've heard of before. It's a position on a chromosome. So you can write a locus like this, like the number of the chromosome, whether it's the long arm or other short arm of the chromosome, and like a particular position on the chromosome. But I think you've got a good grip on what's going on there. All right, and then we have 2 ways to describe the alleles that an individual inherits. If they are homozygous, so zygous reminds me of the zygote. Homo means same. This is when you have 2 identical alleles for a gene. So either they are both dominant or they are both recessive. Heterozygous, on the other hand, hetero means different. This is when we have 2 different alleles of a gene. So something like this. All right, we're also going to run into this word carrier quite a bit, and this is a person who has a recessive allele in their genotype, but because it's recessive, they don't display it in their phenotype. Okay, so they have to be heterozygous, a dominant and a recessive allele, okay, and so they may carry, or we say that they carry this recessive allele because they don't show that trait, but they still have that in their genes. All right, and then a test cross. So when we say cross, we mean like mating or breeding, and test is literally to test something. So these heterozygous individuals are really kind of tricky buggers because if something has a dominant phenotype, like in pea flowers, purple is dominant, okay? And so if I see a purple flower, I don't know whether it's homozygous dominant or heterozygous because they both look purple. So the only way to figure that out is to cross it with a known homozygous individual. So I would take then a white flower and cross it. And if all of their offspring turn out to be purple, then it must be that my original unknown individual was homozygous dominant. If half of them turn out to be white, that must be that my unknown individual was heterozygous. So it's a way that we can figure out the genotype of an individual that has the dominant phenotype. All right, so most of you have seen Punnett grids before. So Punnett grids for a monohybrid cross, crossing one gene or just looking at one gene at a time, looks like this, okay? And up at the top here, we're going to put parent alleles, okay? Here, here. Across the other side here are going to go the other parent alleles, here and here. Okay, and in the middle here, in these boxes, these are going to be the allele combinations of the offspring. I'll show you more about that in a little bit. So if I have a parent who has this genotype, okay, I'm only going to put one, okay, next to each box, either across the top or along the side. And that's because those parent alleles are of the gametes. So that is where the alleles of the gametes go. Okay, so that's because it represents gametes who are haploid. And so when we say haploid, we mean one allele for each trait. All right, so let's talk penguins. In penguins, the pigmentation on their feathers is dominant and albinism is recessive. So we're going to use big A and little a for those. So I'm going to cross a heterozygous male with an albino female, and I want to know what the ratio of phenotypes is going to be. All right, so first I need to determine the genotypes of the parent. So the male is heterozygous, hetero meaning different. So he has to have a big A and a little a, one of each. That's what it means to be heterozygous. And the female, since she is albino and that's recessive, the only way to get a recessive phenotype is to have two of the recessive alleles, homozygous recessive. All right, step 3, set up a Punnett grid to represent the parent gametes. So I'm going to draw my Punnett grid. I'm going to write the female across the top and the male across the side. It doesn't really matter where you write them, which one goes where, okay? And then I want to fill it in to represent the fertilization process. So what that looks like is this baby will inherit one allele from the dad and one allele from the mom. And I want to continue crossing those until my Punnett grid is filled in. Then I want to determine the ratio of the phenotypes. Okay, so I know I have two phenotypes. I either have pigmented feathers, okay, or I have albino feathers. So pigmented feathers, I've got, if I'm going to do that, I've got 1, 2. These are both pigmented, and these albinos are right here. Okay, pigmented because this A, the big A is dominant, okay, albinism is recessive. And I have 2 of these. So if I'm writing a ratio, that would be 2 to 2, or more preferably, I would reduce that to a 1-to-1 ratio. All right, now one of the terms we're going to hear quite often are the F1 generation and the F2 generation. So the F1 generation is the first group of offspring produced by a cross. If I then take 2 of those individuals from that first generation and cross them together, the individuals that I get as a result of that cross are the F2 generation. All right, so let's take a look at this example problem. In pea plants, the gene for being tall is dominant and the gene for being short is recessive. So I know I'm going to probably need a Punnett square here. All right, and I need to figure out the genotypes of the parents. So it says I'm going to take a homozygous dominant plant plant? Well, homozygous means the same. Dominant means dominant. So big T, and I'm going to put that across the top. And I'm going to cross it with a homozygous recessive plant. Again, homo meaning same, and then both the same, both recessive. So I'm going to put those alleles across the side here. And so when I do my cross, I can now answer this question: what percent of the F1 generation will be tall? Well, since tall is dominant, and all of these have a dominant allele in there, that means 100% of the F1 generation is tall. Now let's suppose I want to take two of those individuals from the F1 generation and cross them. Well, all of my F1s were heterozygous, okay? So when I take two individuals from that F1, here's one individual, here's the second individual. When I cross them, I'm going to get a result that looks like this. And now I can answer the question: what percent of the F2 generation will be tall? Well, tall is dominant, so this one's going to be tall, this one's going to be tall, this one's going to be tall, not this one, that has two short alleles. So to answer the question there, what percent is going to be tall? That would be 3 out of 4 offspring, or 75%. So let's revisit this idea of a test cross. So we talked about this earlier. Test cross is when you have an individual that shows the dominant phenotype and you don't know whether they are homozygous dominant or heterozygous, because both of those look the same. So what do? Well, you cross them with a homozygous recessive individual to see what the outcome is and to work backwards to figure out which of these possibilities it is. So here's an example problem: a tall pea plant, and so tall, again, could be homozygous dominant or it could be heterozygous, we don't know, is test crossed, and the resulting offspring are half tall and half short. All right, well, let's consider both possibilities. So I know that I'm going to cross with a homozygous recessive. This is the part about it that makes it a test cross. All right, possibility number 1 is that it's homozygous dominant. Possibility number 2 is that it is heterozygous. When I do my crosses, I can see that this cross yields 100% tall offspring. All of these have the dominant gene, so 100% tall. This cross is going to yield 50%, or 1 out of every 2, tall. So what does that mean? Well, I got to go back into my problem here. It says the resulting offspring are half tall and half short. What must the genotype be? Well, this is the only scenario that matches that information. So the genotype must be heterozygous. Okay, it had to have been that heterozygous plant. All right, now so far I've just been kind of oversimplifying things. So let's back up here. We know that every organism inherits 2 alleles, okay? So we always get 2 alleles, but so far we've only discussed genes that have 2 versions: short, tall, okay? However, most traits have more than 2 versions of an allele. You still only inherit 2, but there are a bunch of different versions to choose from, and that's what we call multiple alleles. And a classic example of multiple alleles is human blood type. Okay, so we have several alleles for blood type, and we write them in a really strange way because they have different dominance patterns. So I'm going to use a capital I to denote that something is dominant, and we have 2 dominant alleles here. The allele for type A blood and the allele for type B blood are both dominant. The allele for type O blood is recessive, and I just write that as a small I. So what we want to do is figure out what kind of combinations I can make here. I can obviously do two type A's. All right, so I could have homozygous A. I could have homozygous B. I could have, well, I better do this correctly. I could have homozygous O. Now I can start mixing and matching these together. I could have an A with a B. I could have an A with an O. Let's see, I could have a B with an O. And I think that's about it. I don't think I can make any other combinations here. All right, so here are the possible allele combinations. See, this is what I mean. Each person only inherits two alleles, but because there's more than two to choose from, we can get quite a few different combinations here. So let's talk about what kind of phenotypes these people are going to have. So these are the genotypes, the actual alleles. The phenotype would be their blood type. All right, so this person would have type A blood. That's easy. This person would have type B blood. Easy. This person would have type O blood. See, it's a lot easier when both alleles are the same because you can tell what they're going to be. All right, this person is in an interesting scenario because A and B are co-dominant. They're both dominant, and co-dominant means they're both going to show up in the phenotype. So this person is going to have type AB blood. So it's pretty neat. Their blood cells are actually going to have the characteristics of both the type B blood and the type A. All right, so this person here has a dominant A and a recessive O. Since O is recessive, it's masked by that dominant A. So this person is going to have type A blood. And the same kind of situation here, dominant B, recessive O. So this person will have type B blood. The only way to get type O blood is to have two recessive alleles. All right, so let's look at a problem involving blood type. So I have a mother with type AB blood who has a child with a man that is heterozygous for type B. Okay, so heterozygous. Well, let's set up my Punnett square. All right, and I'm going to do the mom across the top. The only way to get type AB blood is to have an allele for A and an allele for B. So when she's forming those gametes, half of her gametes are going to have a type A gene and half are going to have a type B. All right, the dad is heterozygous for type B. All right, heterozygous means they have to both be different. So two B alleles is off the table. His genotype has to be one for a B and one for the O. That's the only way to have two different alleles and still end up with type B blood. So half of his sperm are going to have the allele for type B blood and half are going to have the allele for type O. So let's do our crosses here. All right. And when we do that, we can then go back and figure out the genotypes. So this person, well, these are the genotypes. I can figure out the phenotypes. This person is going to have type AB blood. This person is going to have type B blood. So is this person, even though they're heterozygous. And this person is going to have type A blood. So I want to kind of write those out here because it's asking the chances of each blood type for the resulting child. So I have type B. Let's just do that first. There are 2 individuals. So 2 out of 4 is 50%. Type A is just this one. So 1 out of the 4 or 25%. And the type AB blood has a 1 out of 4 chance of showing up, or 25%. Now, with our knowledge of genetics, we can do a much better job of predicting incidences of genetic diseases. And so we're first going to track what's called an autosomal recessive disease. So you know what recessive means, okay? It means not dominant. Autosomal means one of the 22 pairs of chromosomes that excludes the sex chromosomes, okay? So some kind of recessive allele on a chromosome that's not a sex chromosome. How can I tell that something is an autosomal recessive disease? Well, it affects both genders equally. We'll get to gender discrimination in genetic diseases in a little bit. So some common examples here, albinism, so lack of pigmentation, cystic fibrosis, which we've talked about before, phenylketonuria is an inability to digest and break down an amino acid. Sickle cell disease, which we've talked about before, and I know my picture is covering this up here, but this is Tay-Sachs disease. Okay, and Tay-Sachs disease really kind of becomes prevalent in early childhood, and we get a degeneration of brain tissue here from a buildup of pressure. And perhaps we'll talk more about that later in class. All right, so let's do an example here using cystic fibrosis. So here I have two parents who are both carriers, and we know carriers don't have the disease, but they have, well, that's a really bad letter to choose, okay, but they have the recessive allele hidden within their genotype. So I'm going to do a Punnett square here. Okay, assuming that both parents are carriers. Okay, and when I do my cross, then I can start to answer the question. It says, what is the probability that they have a child who is also a carrier? All right, well, see, this person has the disease. They have two copies of the recessive allele, so they're actually not a carrier. This person doesn't even have the recessive allele, so they're not a carrier. My two carriers are here and here, so they will not actually have cystic fibrosis because of this dominant allele, but they will carry that recessive gene. So that would be 2 out of 4, or 50%. Now, genetic diseases, although we talk about them a lot, are relatively uncommon, really rare. Even the most common of genetic diseases affect only about 1 out of every 2,000 people. A typical genetic disease is going to be about 1 out of every 10,000 people, and with some others being much more rare. And that's because people with genetic diseases tend to survive to reproductive age less often. Okay, so like this child here with Tay-Sachs probably won't make it past the age of 5. So he won't be able to pass along those genes. Okay, so the only way for people to get these genetic diseases usually is for both of their parents to be unknowing carriers. And so if you're a carrier, then as we saw, we only have a 1 in 4 chance of having someone with the disease. Okay, but people that have 2 copies of the gene generally don't survive and reproduce. So those genes are effectively weeded out of the gene pool through that method. Okay, so let's move on from autosomal genes to talking about some genes that are on the sex chromosome. So the sex chromosome is right here. It's our 23rd pair, and depending on whether you're a male or a female, you're either going to have 2 X's if you're a female, or an X and a Y if you are male. So what's interesting about the X chromosome is it's a lot bigger. So here's the X chromosome, here's the Y chromosome, and not only is it physically bigger, but it contains a heck of a lot more genes. So thousands of genes on this X chromosome. Some examples include the gene for color vision, clotting factors, growth factors, a whole lot more. Here on the Y chromosome, we're talking about sperm development, testes development, etc. So here it's tough to call these homologous chromosomes because they're really not. They don't contain the same genes. Okay, they just happen to determine our gender. Sex-linked genes were first discovered because when doing genetic crosses, it was noticed that it affects genders in much, much different proportions. Okay, so some of the genetic diseases that we're going to discuss are found on the X chromosome. And if something is found on the X chromosome and it doesn't have a corresponding spot on the Y chromosome, that means that a male is only going to inherit one copy of that trait. So here's what I'm talking about here. The X is a lot bigger than the Y. If the gene for color vision is up here and there's no corresponding gene on the Y chromosome, that means if you're a male, you're only inheriting one copy of that gene. Whereas if you're a female and you get two X's, you inherit two copies of that gene. So if I inherit some kind of recessive gene on one X chromosome, but I have a dominant gene on the other one, then I'm going to display the dominant phenotype. This one will be masked. Unfortunately, if you're a male and you receive a recessive gene on one X chromosome, there's nothing on the Y chromosome to counteract that. And if this recessive gene codes for genetic disease, you're going to get that. And some of these diseases include color blindness and hemophilia. So color blindness, the inability to distinguish between colors, there's lots of different types, and hemophilia is the inability to produce certain clotting factors, so excessive bleeding can be very dangerous. All right, so again, color blindness is the inability to distinguish between certain colors, okay? So there are many different types of color blindness, but as you can see, it affects males in much, much, much greater proportions. Again, that's a recessive X-linked trait. Okay, and hemophilia is also recessive and X-linked, and again, it's unable to produce certain clotting factors, so you end up bleeding very easily. And back in the 80s, like when I was a kid, we often heard of hemophilia patients contracting HIV. That's because hemophiliacs tend to need a lot of blood transfusion, and back before AIDS was well known or able to be detected, unfortunately, a lot of these individuals ended up receiving blood transfusions from people that were carrying the HIV virus. So some really kind of interesting history involved with this disease. All right, so let's talk about color blindness some more. So we know it's X-linked. So for the alleles for color blindness, oh, that was really shaky. I can either have an X with a capital C, and that's the dominant regular color vision, or I can have an X with a small c, and so that would be the recessive color blindness. And then, of course, the Y chromosome does not have any gene for color vision. So let's talk about some genotypes. All right, so I can have X big C, X big C. I can have X little c, X little c. Those would be my two females, and there's a third female possibility, X big C, X little c. All right, and then for my male possibilities, I could have X big C, Y, or X little c, Y. All right, so let's talk about these phenotypes then. So this is going to be a female, XX, okay, with regular vision, because regular vision is dominant. This would be a female who is colorblind, okay, two recessive alleles. And here's a female, so XX, female, and she has one dominant and one recessive gene. So since regular vision is dominant, she has regular vision. All right, over here, XY, we have a male, and since he only has one copy for color vision and it happens to be dominant, the regular color vision, he is going to be a male with regular vision. And over here we have a male, and again, he only has one copy of the X chromosome, and it happens to be recessive for color blindness, and so he's going to be colorblind. So here's where things get interesting in regards to carriers. So remember, carriers have one copy of a recessive allele, but they don't show that recessive phenotype. So females can be carriers, like this female is a carrier. She carries that recessive allele, but she's not colorblind because she has this dominant gene for color vision, or for regular vision, yeah. So males, on the other hand, can't be carriers. To be a carrier, you have to have the recessive allele, okay? And since the Y chromosome doesn't have that gene on it, this person is going to be colorblind. So that doesn't count because, remember, carriers don't show the recessive trait. Okay, so let's do an example problem here. Okay, in this case, we're going to be talking about hemophilia. Hemophilia is also X-linked, and you can either have the dominant allele for regular blood clotting or the recessive allele for hemophilia, but remember the Y doesn't have any gene for that on there. So I'm going to draw my Punnett square. Okay, and it says a man with hemophilia. Okay, so a man with hemophilia is going to be XY, and if he has hemophilia, he must have the recessive hemophilia gene on his X chromosome. Has a child with a woman who is a carrier. So carrier means one dominant and one recessive. And so what's the probability that they have a child with hemophilia? Well, if I do my cross, that'll help me determine that. Okay, so it's just asking for a child. It's not asking for a specific gender, so I can consider all four of these possibilities. Let's find the ones that have hemophilia. This one doesn't. That dominant, capital H right there, tells me that they clot normally. Same here. Okay, but these two both have hemophilia, so that would be 50%. Okay, so this one I think you can do on your own, okay, if you really think about it. So I want you to think about how men get color blindness, like how many alleles it takes to do that, and how women get color blindness, how many alleles it takes for that to happen, and then consider why way more men, like this represents a ratio, like 1 out of this many, versus for women, 1 out of this many are colorblind. All right, so we're going to take our knowledge to the next level by using what we've already learned about genetics to interpret these charts called pedigrees. So pedigrees involve a fair amount of symbolism. So a square is a male, and a circle is a female, and anything that's shaded in usually means they are affected by something, whether it's a disease or they have a certain trait or what have you. Vertical lines show parent to offspring relationships, so parent to offspring. So here, this individual is connected by a vertical line to these parents. This is his parents. A horizontal line equals some kind of baby-making partnership, okay? Usually you'll hear that in pedigrees referred to as a marriage. That's kind of old school, okay? So these people share a horizontal line. That means that these 3 individuals are their children. Okay, so we're going to work with cystic fibrosis first. So cystic fibrosis is an autosomal recessive disease, okay? So I don't have to worry about males and females, X's and Y's. I'm going to give this a capital C, and that's going to equal the dominant allele for like regular lung function, or a little c, which is the recessive allele for cystic fibrosis. So the easiest ones to do are always the ones that are affected, and that's because they must have 2 copies of the recessive alleles. Okay, now I have to work backwards. I think it's easier to start from the bottom and work our way up. So this person has 2 recessive alleles. Well, where do you get your alleles from? You have to get them from your parents. And since he got 2 of them, 1 had to have come from each parent. So each of these parents has to have a little c somewhere in their genome. Now, since they don't have the disease, that means that they must also have a dominant allele, okay? So they are both carriers. Now, these individuals down here don't have the disease, so they have to have a copy of the dominant allele. But what I don't know is what their other allele is, because this person could have gotten a big C from one parent and a small c from another parent and still been unaffected, or she could have gotten a big C from one parent and a big C from another. So unfortunately, we don't know what the other genotype is. And I guess I shouldn't write it like with the question mark. She can either be big C, or big C, little c. And same with this one. Okay, we just don't know those. Alrighty. Now let's take a look at these individuals over here. So we know that this parent has to have at least one big C in his genome, okay, and that's because he doesn't have the disease. I don't know what the other one is just yet, so let's work on their children. All of these children have to have at least one big C because none of them have the disease. They also have to have another little c, and I'll tell you why. This female can only donate little c's to her offspring, okay? So all of these had to have gotten a little c from their mom. Okay, so again, there's nothing here since we know the little c comes from the mom and the big C comes from the dad. We don't know if this dad is big C, or big C, little c. Okay, it's probably most likely that he's big C, because if he was big C, little c, then perhaps some of his children would have inherited a little c and had the disease, but we don't know that for sure. All right, now let's take a look at a pedigree that involves genes that are co-dominant. So you're going to notice in co-dominance, we don't use like big letters and small letters. We use two different letters because they're both dominant. Okay, so in this particular pedigree, individuals that are completely shaded are going to represent the red cows. So in order to be red, you have to have two big R's, okay? Because if you have one R and one W, you end up with this color called roan. So a lot of times in pedigrees for codominance, they will half shade a circle to represent something that is codominant. So here are my codominant individuals. Okay, they are roan, they show both the red and the white spots, and then the rest of them have to be just white, okay? Because the only way to be white, if you are this kind of cow, is to get two white alleles. All right, so let's take a look at a sex-linked pedigree, and we're going to use hemophilia for an example. So remember, hemophilia is found on the X chromosome and nothing on the Y chromosome. So this would be the gene for hemophilia. And the easiest thing to do is go along and get the genotypes of all of these males with hemophilia. So all of them are going to have that recessive copy on the on the X chromosome, and then they're going to have just a normal Y, because there is no copy, or gene rather, for hemophilia on that Y chromosome. All right, now let's do some backtracking. So I know Leopold here had to get his Y chromosome from his dad because mothers can't donate Y chromosomes. So the Y comes from the dad, obviously, meaning that this recessive X chromosome had to have come from Victoria, the mother. And she doesn't have hemophilia, so her other chromosome must be the dominant one. Albert also doesn't have hemophilia, so he must have the dominant version also. All right, next thing I'm going to do is I'm going to take a look at all of the males without hemophilia. They have to have a dominant gene on that X chromosome, because again, there is nothing on the Y, and if they don't have hemophilia, that means the only copy they have must be the dominant for normal blood clotting. So I'm going to go through and fill in all of those. Let's see, am I missing any here? Okay. Now I'm going to go ahead and stop here. This pedigree isn't the best for determining all the genotypes because it neglects to show us the other partners involved in some of these children. So we're going to go ahead and leave those alone, but I'm hoping that you're kind of getting the gist here as to how to backtrack and figure out which chromosomes had to have come from which parent. Okay, so we know that mutations in the DNA can cause genetic diseases, but they can also cause other things like cancer. So mutagens, that's a word you need to know. Mutagens are something that cause a change or a mutation to the DNA, and that could be a whole host of chemicals. It could include things like UV radiation or other types of radiation. There are lots of possible mutagens, and radiation is interesting. It can cause damage to the sections of your DNA that regulate cell division. So this is Madame Curie, one of the great woman heroes of science, and she actually died of ovarian cancer, probably due to the radiation that she was exposed to in her lab. So you have sections of your DNA that are supposed to turn on when cell division is necessary and then turn off when cell division isn't necessary. And for some reason, these mutagens kind of keep that in the on position, and they just keep dividing out of control. Another possible source of radiation includes X-rays and nuclear radiation. So this is one of the radioactive boars currently inhabiting the space where the Fukushima nuclear power plant was in Japan. And these guys have been eating all of the vegetation and insects and stuff that have been exposed to the radiation, and now they have high amounts of radiation, and their incidence of cancer and other genetic mutations is incredibly high. So all of those are due to these mutagens. Okay, so let's say that I'm a woman carrying the trait for sickle cell disease, and I decide to have a child with a man who's who is also a carrier for that sickle cell trait. If I do a Punnett square with our genotypes, I should be noticing that about 1 in every 4 children should have sickle cell disease. But I have 4 children and all of them have sickle cell disease. How can that be possible? Well, we have to remember that these Punnett squares are predictors, okay? They are not guarantors. So what they do is they calculate the genetic probability, and probability is not a guarantee. So if I were to have 1,000 babies, I would expect 25% of them to have sickle cell trait. But with such a small sample size of only 4 children, it's possible that I might get outcomes that are different than what I expected. Now, it's really important that we understand where these genetic diseases arise from, okay? And so a lot of funding goes into searching through our entire genome for those genetic diseases, okay? However, studies that are just trying to understand the functions of other genes not related to diseases largely go unfunctioned. And that's all because of money. So if you find a gene that happens to control a genetic disease, you can make money off of then finding cures or more information about that disease. But if, let's say, you find a gene that just controls how a certain protein in a cell is made, wow, that's really nice to know, it doesn't have any money-making capability. And so funding for that largely is just forgotten about. All right, now just because you do research on something doesn't mean that everybody's going to completely change how they're doing anything. And a great example here are some studies done back in the 1950s that looked at x-rays and the effect on the children from pregnant women who were exposed to those x-rays during pregnancy. And they found that those children, again, of the women who were x-rayed, had greatly increased risks of cancer and other diseases later on in life. And so even though that research came out in the 50s, it wasn't until the 70s when doctors finally put an end to x-raying pregnant women. And that's because we, as a scientific community, really want experiments to be repeated. We want large numbers of data. We want to make sure that the results that we are getting aren't anomalous. And so sometimes that can take quite a long time. And that'll just about do it for chapter 3.4 on inheritance.